The overall goal of this model is to create a clinically relevant neurotrauma phenotype in a 96-well format to facilitate the efficient and simultaneous comparison of injury sustained under multiple experimental conditions.
The primary purpose of this model is to enable high-content phenotypic drug discovery screens in neurotrauma with human-induced pluripotent stem cell-derived neurons.
The plates used in this model retain the industry-standard 96-well plate geometry, maintaining their compatibility with existing high-content screening machinery like robotic arms and automated microscopes.
To begin, place a bottomless 96-well plate bottom side up inside a 200-watt plasma cleaner for 60 seconds on high power. Next, place the plate in 200 milliliters of freshly prepared 1.5% Apdes in deionized water, bottom side down, for 20 minutes in a fume hood. 10 minutes into the incubation, use forceps to position a plasma-treated silicone membrane on top of a 7.5 by 11 cm parchment paper rectangle and align a 7.5 by 11 by 0.5 cm aluminum slab on the lower portion of the plate fabrication clamp. Then use forceps to align the silicone membrane and parchment paper on the aluminum slab. At the end of the coating period, remove the bottomless plate from the Aptes bath and shake off the excess solution. Dip the bottomless plate into 2 consecutive 200 milliliter deionized water baths for 5 seconds each dip and shake off the excess water. After complete drying with compressed air, place the bottomless plate into the upper portion of the plate fabrication clamp and gently close the clamp to press the bottomless plate and silicone together.